Hello, welcome to this Practicum lecture. This is a pre-lab lecture for a tutorial for bioinformatics. So the objective for this entire activity lecture lab itself is to introduce some commonly used databases and bioinformatic tools for analysis of DNA, RNA, and protein, and to illustrate the flow of information, which is the central dogma in cell biology, that the information goes from DNA to RNA to protein with a medically relevant example. So the first tool that you will be working with is the NCBI gene database. So this will give you general information about the gene, and some of it is like the name, alternative names, an accession number, so a unique number, an ID number for that entry for that gene, and organism, lineage of the organism, and things like that. It also gives you a written summary of what is known or the relevance of that gene, and some sources that you can look up in case you want to know more information about it. You also, as you scroll down, get a little bit more information. For example, the chromosomal context. In this particular is the COX-1 gene or PTGS1, and the location here is 9q32.2. It's really the 9th chromosome. Q is the arm of the chromosome that is the longest, is traditionally named as Q, and the shorter arm would be P. And then 32.2 is like a banding on the chromosome. Once you identify your gene, you want to look up the RNA sequence. And that's, we're looking for a reference sequence. So multiple versions of the gene that has been identified sometimes. And so this database, RefSeq, finds the data, the sequence that seems to have the most valid data, the most supporting data, and then defines that as the reference sequence. And there might be slightly different variants from person to person, or depending on the techniques used, but this is what they consider to be the most valid and likely the most common sequence for that gene. And then the accession number would have an NM if it's a messenger RNA, or NP if it's a protein sequence. So all of the messenger RNA would start with NM for the accession number. And then once you open the reference sequence page, you would see something like this. So it has the locus, which has the NM number and a unique identifier for that gene, so that if different groups end up naming it different things, we know it's the same gene because it's on the same locus. And then you have the size. So this has 4,000 base pairs. It's a messenger RNA, and the lineage. And then also lists underneath some references that supports that data for that particular sequence. The next thing we will do is to collect the FASTA formatted sequence. So from that original page, you have a FASTA link. It's right below the NCBI, right below the name. And that opens up another page that has the FASTA sequence. So the sequence, it is formatted in a specific format that's recognized by most bioinformatics tools. And that format basically has that title first with the larger than sign, and then the text, and then a paragraph marker, right? So a new paragraph is starting, and then the sequence comes below it without any spaces or paragraph separation. So it's very important to keep all your sequences on this FASTA format. So you want to copy and paste both the title and the sequence, so you have an identifier for each of the sequences. And then you will also look up a protein sequence database. So the UniProt TKB, it is a database for protein, or has information about the protein that's coded by that gene. And so in this case, you have like a function, you have miscellaneous related to function as well. So you have like a pretty decent amount of written information about it, some references, and then if you keep scrolling down, you keep getting more and more information about the gene. For example, it's an enzyme, so what's the catalytic activity of this enzyme, the kinetics of that activity, and I want to highlight that it also lists key featured sites. So a binding site, active site. So a binding site is it's a unique amino acid within the protein that's been shown to interact and bind to the substrate. So that amino acid is required for the substrate to bind, and you would then can assume that removing that binding site then can interferes with the ability of that enzyme to bind to its substrate, and therefore the enzyme activity would be impaired. The active site also has been shown to be required for the activity of the enzyme, and so it's a specific location in the protein that we know it's very important for the function. And then you have a metal binding site, so RN is a cofactor for this enzyme, and this is where it binds and interacts with the protein and this particular amino acid. And you have this other site there, that's the aspirin acetylated site. So it's the amino acid in the reference sequence that gets altered with interacting aspirin. So within any protein, you can find a number of these informations, and you can find some more information that you will be looking for as you go through the worksheet. Once you collect all the background information, we'll move on to part 2, and that's where we'll do a little bit of data manipulation. So XPASY website is like a portal for multiple bioinformatic tools, and this Translate is one of them. So you can then, in this box, input any sequence in FASTA format for either DNA or RNA, and then it will produce the translated sequence. So it will read what that RNA or DNA sequence would code for in the protein sequence. You can also select here different versions. You can do a verbose or a compact, and the only difference there is M versus MET and then a dash instead of stop. I think the verbose is a little bit easier for beginners. It also allows you to define the DNA strand that you're looking for. If you know it's on the forward strand, or if it's on the reverse strand, you can select which strand you want. In our case, let's select both so you can see all of the options. So that means it will translate every single possible product for this messenger RNA sequence that you will input, and that means it will have 6 different frames. So it will have the 5' to 3' direction, frame 1, 2, and 3, and then 3' to 5' direction, frame 1, 2, and 3. And those frames is because it reads the nucleotides in triplets, right? So it starts from nucleotide, the first one and second and third reads for one codon, meaning one amino acid. And if it starts from the second one, it will read a complete different amino acid. And not only the first one, but all of the ones subsequent will also be one amino acid or one nucleotide off, which means will be one amino acid difference. And so you have a complete different coding coming if you start on the second. And the frame 3 is if you start on the third. Of course, if you skip all 3, then you're back to frame 1. You just skipped one amino acid. So you have 3 different frames in one direction and then 3 different frames on the opposite direction. So this tool also highlights in red any segments that would transcribe or be translated into a protein. So you can look into these 3 different versions here. You can see which one seems to be most likely to be an actual protein. It's likely to be frame 2 because it has the most continuous red. Both frame 1 and frame 2 has small segments that could be translated into a protein, but they are unlikely to actually be a functional protein because they're too short. And then for once you have your protein sequences, we'll do a sequence alignment. So sequence alignment, first you want to make sure that you have the correct formatting, that they're in FASTA format, and then we'll use a cluster omega tool where you will paste all of your sequences. You can do a pairwise, meaning 2 different sequences, but this tool also allows you to input multiple sequences and will align all of them. So when it does that, it uses a specific algorithm that then calculates what position do they need to be in to have the most amino acids that are the same or closely related. And you will figure that out and you will align them. So we will do this with PTGS1, PTGS2, and PTGS2 from other species, not just human, but other mammals and other species. So this is the cluster omega tool, and the alignment should look something like this. So in this case, I don't have PTGS1, but we have PTGS2 and the different animals. And that amino acid count is on the far right, and that is the number of amino acid that's the last one on the sequence. So that N there in the last, in the human one is number 53. I'm Without my glasses, I can hardly see it right. So it's the 53, and it's the same 53 in most organisms except the coral. Coral is the most evolutionarily distant organism, so most of the other ones all of the other ones are mammals, and so they are fairly similar. And then we have something quite a bit different, so that we can compare in terms of more evolutionary significant. So you would expect that any those active sites, um, the, 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 the substrate binding site, iron binding sites, that those would remain the same. So even in more distantly related organisms like the coral, we would expect them to be conserved, and therefore the same active sites, the same binding sites would still be present in all of those proteins. So where you have alignment and consistency among all of the sequences, on the very bottom of the line you have like the concordance of all the alignments, and it will show an asterisk there as indicating what is exactly identical in all of the sequences. And if there is no alignment, no matching whatsoever, then you would have nothing. You have no symbol at the very bottom. If you have in any of the sequences a dash, that means there's a gap where it would align. So for example, the beginning of the sequence, the coral protein is a little bit longer than the others, and that is matched with basically nothing in all of the others. So all of the others have little dashes to indicate no amino acids that correspond to those amino acids in the coral. But then you have, the first one has some matching, some homology. Homology just means similar function. And then the second amino acid is identical. So that's why it's aligning that way, as opposed to having gaps in the middle of the sequence. All right, so your job will be just to look at that and then compare follow the worksheet, but compare BTGS1 and BTGS2, what's happening to the amino acid that is altered by aspirin. All right, you have 3 options for your following the worksheets. You can just follow the worksheets and has all the instructions there, and that should be sufficient, especially if you're familiar with the tools. If you are less familiar, or if you have difficulty having too many screens on open, you can print out the lab manual, so you have a paper version and you can write down the answers to the prompt questions, and then the questions then will be the same that are on the worksheet online, and then you can transcribe the answers later. And then a third option is to watch the video. I find for a lot of people it's just difficult to describe what I mean when I'm telling you to do certain things. So if you watch the video, I'm doing exactly what you're supposed to be doing, so you can just do the same thing and come up with the answers for all the worksheet questions. And then either when you finish or when you just need to take a break from the worksheets, I ask you that you then try to do it soon. Go for the self-assessment for mindset. It's a little bit of a metacognition just to give you some insight into the learning process, but do that thinking about the experience that you had working through the worksheets, or the experience you've had working through any task that you found to be challenging. All right, so thank you, and I'll see you in our synchronized session later on.